Hello, everybody. In this lecture, we are going to continue on our eukaryotes, and we're going to start talking about the specific kinds, and we're beginning with fungi. You know these lovable microbes from the mushrooms that you can see growing in the forest or when you buy them in the grocery store. So let's get right into it. By the end of this module, you should be able to identify structural characteristics of fungi and differentiate between unicellular, multicellular, and dimorphic fungi. All right. So when we're thinking about fungi, there are a lot of different species. There are greater than 600,000 of them. Most of them appear in this multicellular form, like we see in these images on the right-hand side. We've got different types of mushrooms, and those mushrooms might be growing out of the soil. They might be growing on decaying like branches or wood, and even on like sometimes on living trees and that sort of stuff too. And so they come in all these different formats. Um, the yeast form of fungi are going to be unicellular. Um, and they are, we saw those in labs. So those are eukaryotic cells. They're nice and big. And then your multicellular kinds can have these fruiting bodies like these fungi. Some of them are just molds, right? So when you see moldy bread, that is going to be a multicellular fungus. Now, these things do not carry out photosynthesis. Instead, they are going to be heterotrophic, which means that they are getting their nutrients from the environment. Um, some of them can be pathogenic, and more even are going to be saprotrophic, which means that they are basically decomposers. So we're going to talk a little bit about structures in fungi. And one of those structures are hyphae. There are two main types of hyphae. We have septate and aseptate. So let's look at a picture of that. So here we have a cartoon on the left-hand side of septate hyphae, and you can see these like, they're kind of like, little hair-like structures or root-like structures that reach out. And the septate hyphae have these little delineations, these septa, between sections with their own individual nuclei. Now, with your and we can see that in this image below, where we can see these septa are being highlighted by the arrows with these lines. And then we have aseptate hyphae. And then the aseptate hyphae, you have multi nuclei in one space, right? So there are a bunch of different nuclei, and they're not separated by these, different cells, right? The cell wall is just on the outside. We're not seeing anything separating these subsections between. And you can see that down here, there are no septa. Now, as I said, most fungi are multicellular, and that's going to be your mold form, which also is what results in our fruiting bodies, which are the mushrooms. And then our unicellular fungi are going to be called yeasts. So this is the same sort of thing that we make bread with, which is Saccharomyces cerevisiae, or some of the yeasts that can cause, infections as well. Now, the kind of interesting thing is that some fungi can exist in what's known as a dimorphic state. And what that means is that under um, certain conditions, they grow as molds, and as under other conditions, they grow as yeast. And so the big example for something like this, if we look at this image, um, you can have temperature dependent dimorphism, which means that under the right temperature, you take a yeast form, and not in that temperature, you have a mold form. And so a true mold that is just a mold, if we grow them at two different temperatures, it's going to be a mold regardless. It produces little mycelia, and they give you that characteristic mold format that looks kind of like fluffy. Oftentimes, it might be green or black. It's kind of stuff you see on bread, cheese, etc. Now, your yeast, they grow more like a bacteria A true yeast is going to grow like a bacteria, regardless of the temperature. Now, a dimorphic fungus, on the other hand, is going to grow like a mold under some temperatures, and under other temperatures, they grow like a yeast. So the classic example here is you have the environmental form is the mold, and then the pathogenic form is the yeast. So if you grow it at a low temperature, like 30 degrees Celsius, then you're just going to get mold. But if you bring it up to body temperature, which is 37 degrees Celsius, you get the yeast form. And that's where you can have some pathogenesis. Fungi can produce spores. These spores can be both sexual and asexual. And here are several different, images to explain, like, or to kind of visualize how these spores can exist. And so they come in these formats of, or in our asexual spores come in these different formats. We have conidiaspores and sporangiospores. And your conidiaspores are going to be more like these little individuals that are coming off the ends of these conidia. And then your sporangiospores are going to be in these little sacs like this. And so all these spores are encased in this one big sac. Um, and so these asexual spores, right, they're being produced through mitosis. And so they are just going to be complete genetic replication or duplicates of the mother cell, right? So the spores aren't going to have genetic variation between them, and they are going to be produced during the mitotic division and not the meiotic division. Um, Now, on the other hand, you have your sexual spores, and the sexual spores are produced during meiosis. And since they're produced in meiosis, they are going to have genetic variation. And you've got a couple of different types of sexually produced spores, including your zygospores, ascospores, and basidiospores. The important thing to kind of know between these two is I'm not really super worried about you having a detailed understanding of or memorizing the differences between sexual and asexual spores in terms of like the conidiaspores and the zygospores and that sort of thing. What I'm more interested in is you understanding how a asexual spore is different from a sexual spore, and including like what process produces them. And here's a nice little um, comparison of different types of spores. This is can be fun to look at if you're interested in learning little bit more about some of these structures and differentiating them. But in terms of just like what I want you to think about for class purposes is more along the lines of, you know, what are the haploid ones versus which are the diploid ones. It doesn't say diploid over here, but our asexual spores should be diploid. And what are they being produced for? So our sexual spores are going to allow for sexual reproduction, while our asexual spores are going to be used for basically making clones of yourself, letting them grow somewhere else. All right, so we can have diseases that are caused by fungi. We call these diseases mycoses. And then we've got a couple of options where mycoses exist. One of the most common ways is if you're immunocompromised. This is going to be stuff like pneumocystis pneumonia that AIDS patients get. So this is uncommon to get this kind of pneumonia if you're healthy, but if you have a suppressed immune system, then it's fairly common. Um, Also, when you get some sort of disruption of your normal microbiota, you can get fungal infection. So a common one here is a yeast infection. When you have an issue with, say, the vaginal microbiota, your healthy lactobacilli are gone or have reduced numbers, and then your Canidia yeast can come in and actually cause infection. There are other true pathogens that exist within the group of the fungi, and these are going to be things like histoplasmosis or coccidiomycosis, and both of these cause disease in healthy individuals, and we see these different skin rashes that come from them. Then there are a lot of dermatophytes. These are the ones that you are likely at least somewhat familiar with. We've got things like tinea pedis, which is athlete's foot, right? You've got that can result in like obviously itchy areas and agitated skin. You can also get these like nail infections, these nail bed infections. And then you have mycotoxins. Mycotoxins are pretty important because these are toxins that are being produced by a fungus. So that's what if you haven't caught on to that yet, the myco prefix is a prefix meaning fungus. And so um, we've got Claviceps purpurea, which produces the ergot toxin. This is a neurotoxin that can cause things like seizures, psychoses, nausea, vomiting, and even death. And so this is one that you probably have heard of, at least in some extent, directly or indirectly, because these mycotoxins, like the ergot toxin, is one of the things that is thought to potentially have led to the Salem, witch uh hunts, right? So you had these young women who potentially were poisoned by ergot toxin, which can be found on rye. So you have these rye houses, and you can get the growth of this fungus on the rye. And if you eat that, you can get um, you know, this neurotoxic effect that results in things like psychoses and seizures, which are so it's been proposed as one of the things that could have been happening to those young women in Salem. All right, with that, we only actually did the top two. You should be able to identify structural characteristics of fungi and differentiate between unicellular, multicellular, and dimorphic fungi. With that, I hope you enjoyed, and I'll see you soon.